everyone, welcome to our second video about the scientific method. In this video, we're going to talk about a couple different things: observations, inferences, predictions. We're going to review parts of the experimental design and talk about data and writing procedures. So get ready for that. Observations, inferences, and predictions. An observation uses your senses, just like you learned in the previous video. You're going to use your sense of sight, sound, taste, touch, and smell. Inferences use observations, prior knowledge, or logical reasoning to come up with an explanation for a past or present event. Basically, an inference is going to be you're drawing a conclusion. And a prediction is thinking about what might happen in the future based on what you observed or what you already know. Here's some examples. I want you to think, is this an observation, an inference, or a prediction? If I add water into a container of strong acid, it will explode. Now, this hasn't taken place yet. We're saying it will explode, which is in the future. So this is a prediction. Next, the dog was crying. He must have been scared of the storm. We're coming to a conclusion about this dog. So it's an inference. Lastly, if the student has 3 A's and 4 B's, we're making a observation because that is a numerical piece of data, like 3 A's and 4 B's. That's actually a quantitative observation. Quantitative means quantities, numbers. Review of IV, DV controls and constants. Now, we talked about this in the last video, but it's very important, so just make sure that you are aware that the independent variable is the one that you, as the experimenter, change or manipulate. I change the IV. It's also usually next to the word if in the hypothesis. So if your independent variable, then your dependent variable. If I grow plants in different liquids, then plants will grow the tallest when watered with water. So the IV is the type of liquid. If I water plants with different types of liquid, the DV is how well the plants will grow. It might grow taller with a certain liquid. Dependent variable is the variable you want to observe. It's the responding variable. The data comes from the DV, or the dependent variable is measurable. If you see the DV in the hypothesis, it's usually next to the word then. So if IV, then DV. The control is the group that would get the normal treatment. A lot of times that's going to be considered water, especially in experiments in a biology classroom. Constants are the factors that you keep the same for each group in the experiment. Those are the things that help us remember that we only want to change one thing. We only want to manipulate one variable, and that's the independent variable. We want to keep all things other than that one thing that we're trying to test the same. All right, here's an example. Seth Spartan noticed that he runs faster when he drinks Gatorade instead of water. He gives 100 student athletes to help test the hypothesis that if you drink Gatorade, you will run faster. 50 students drink 1 liter of Gatorade before running a mile, and 50 students drink 1 liter of water before running a mile. Then Seth compares the average running time. See if you can think of what the IV, DV, control, and constants would be in this experiment. The independent variable is what kind of drink the runners are having. So is it going to be Gatorade or is it going to be water? There are 2 levels of the IV here, Gatorade and water, but there's just one thing that the experimenter is changing, what type of drink they're having. The dependent variable is the running time, how long it takes for the person to run a mile. The control group is water, because water is considered the normal thing that people might drink. And the constants would be you would want to have these runners run at the same day, at the same track, around the same time, drinking the same amount of fluid, and having the same distance to run. You want to keep everything except what type of drink they have the same. So all those other things would be constants. When you're doing an experiment, you want to make sure you have a solid procedure to work with. That's important for you as the experimenter, but it's also important for others if they're trying to retest or repeat your experiment. Your procedure needs to be step by step. It's got to be really detailed, complete, and clear. A stranger should be able to read it and understand exactly what you mean. So, I'm going to give you some examples of bad and good procedures. I want you to think about why they're bad or good. Um, when you're thinking about it, think about how to write a procedure. You're going to list the equipment, you're going to list the steps in order. You're going to be clear and precise. You're going to be short and sweet, not too wordy, but making sure that you give enough details and start each step typically with a verb. This is a bad example of a biology procedure. Get a jar and some lightning bugs. Put the bugs in the jar and count how many times the bugs flash in 3 minutes. Heat up the bugs by putting them near a heater. Count how many times they flash in the next 3 minutes and record results. What's bad here and what's good here? Well, you don't really know how many bugs, what kind of jar, how many bugs are in each jar, how big is the jar, how hot should you make the air next to the jar, and how many times should you repeat the test. It's hard to read because it's not in a bulleted format either, but it does have some good aspects. It is talking about a certain set number of minutes, but it's really not too detailed. This is a much better option for a biology procedure. You can see on the left that the different materials being used are listed and the steps are there, and they're much more detailed and precise. They're talking about specific temperatures and a specific number of bugs in any given jar and how to repeat the steps of the procedure. Next, I'm showing you a bad chemistry procedure. Put sodium bicarbonate in a test tube, then put hydrochloric acid in a test tube. Observe them and record your data. What will happen when you mix it? Mix the substances, observe it, then clean up. Well, you might be thinking there's some bad here and some good here, but some of the bad is how much sodium bicarbonate? Is it diluted hydrochloric acid? How much hydrochloric acid? Should I write observations before mixing the substances? Should I add sodium bicarbonate to hydrochloric acid, or should I add the hydrochloric acid to bicarbonate? How should I clean it? A better chemistry procedure, well, you might want to also include the materials that you'll be using, but here's a better example of how the actual procedure is written. Place a small scoop of sodium bicarbonate in the test tube and 5 milliliters of dilute hydrochloric acid in another test tube. There you have more details about how much and what kind of material you're using. Observe the two substances. Record your description in the data table. Predict what will happen when they are mixed and record your prediction. Carefully pour the acid into the sodium bicarbonate, so now you know how you're going to be mixing it. Observe and record what occurs. And then finally, when the reaction is finished, pour the contents down the sink and rinse out the test tubes. Now you know how to clean up. When you finish an experiment, you're going to do some data analysis and you're going to need to know some keywords: range, median, and mean. The range is the difference between the highest and lowest data points in a given set. So in this case, the range is going to be 9 because 14 minus 5 is 9. The median is the middle data point. You can put the numbers in order and cross off one from either end until you get to the middle and find out here that the median is 9. The mean is the average. You can add up all your data and then divide by the number of data points to find your mean. Here's an example with some data. You could take some time to calculate the range, median, mean for this data, but I'll go ahead and show it to you and tell you how I would have gotten that. So for the range, I would subtract the highest number minus the lowest number to get the range. The median would be the middle number if you put them in order from smallest to largest. And the mean is if you add all the numbers up and divide by the number of data points, you would get that. So in this case, you would add 5 plus 4 plus 2 plus 5 plus 6, and you would then divide by 5 because there were 5 data points. Here's a difference between two important words: precise and accurate. Precise means how close are two measurements to each other. Basically, if you repeated the experiment, how close are your results? Accurate is how close it is to a predicted or standard or known value. So take a look at these little targets to learn a little bit more about precise and accurate. In this case, you have low accuracy and low precision. It's low precision because the data is not close to itself. It's not accurate because it's not close to the center of the bullseye, which is the known value. In this case, you have low accuracy because it is not close to the center, which is your known or expected value, but you have high precision because all the data points are together. In this example here, you have high accuracy. Most of your data points there are close to the bullseye, which is the known value, but you have low precision because your data points are a bit spread out. Finally, over here, you have high accuracy and high precision. It's high accuracy because all the data points are right at the bullseye, the known value. It's precise because all the data points are right next to each other. Thank you so much for listening.